Okay, so last but not least, I'm going to conclude the session this morning. And what I want to take a look at is really the role of the microbiota in enteric infectious diseases, diarrheal-type bacterial-mediated diseases. And I think Bill showed you already the microscope that Anthony van Leeuwenhoek used to actually examine the microbes in his mouth, and he came up with a very profound statement a long, long time ago that there's a lot of microbes out there, even in our mouth, and in this case more people in his mouth than in the Netherlands at the time. And of course I talk about diarrhea, so there's the warning before lunch. Okay, I'm not going to spend a lot of time on this, but just to remind us all that there's varying microbiota composition in and out all over our bodies, and as you go down the intestinal tract, basically you get more and more till the colon, where there's phenomenal numbers of bacteria in there. And I think we've just had a really interesting discussion on how you analyze these things, and it depends what level you look at where you can find similarities and differences. If you're very high up on the classification system, when you're looking at the phyla level, for example, in the gut, basically mouth and human actually have a moderately similar type distribution, and the vast majority of bacteria in both these intestines are the firmicutes and the bacteroides, with a little bit of proteobacteria thrown in. So, and we will be talking more about this and the changes in this during enteric infection. So this goes back several years now in our lab. We had a lab retreat, and one of the many things we discussed was this question. It sort of came up just in a free-ranging discussion we have, and relatively simple question. And I guess I'd always assumed it was well known. People have been studying diarrhea and what's in the feces, etc., for a long time. But when we came back and had a look, and this is Claudia Lupp that really originated this work, came back and had a hard look at the literature, it really wasn't very well known at all. Partly because the techniques weren't there, and perhaps partly because it just hadn't been done. So that's kind of where I want to pick this story up and start in on showing you what actually does happen to the intestinal microbiota during diarrhea. So, as many of you know, we usually work on pathogenic E. coli and Salmonella in our lab, and we wanted to study a model that would allow us to really analyze the microbiota when these animals have these enteric infections. And it turns out that in studying the E. coli in these mouse, there's a very close relative to the E. coli, something called Citrobacter rodentium. It's not called E. coli, but it's actually a terrific mouse model for the diseases caused by enteropathogenic E. coli and enterhemorrhagic E. coli, or E. coli 157. And this mimics the disease where the bacteria attach and efface in these cells and has all the same kind of virulence factors of these things. And we've been using this mouse model for a long time to study the infectious process. But the nice thing is this is a natural occurring infection. So this is not an animal model. This is a natural pathogen of mice. And you can see it's so named because it's a mouse pathogen. And so here we have a natural pathogen and then mice to work with. So that's where we started the story to see what would happen. So when you infect mice with the Citrobacter rodentium, what happens is that basically it's a span over 4 weeks, the way the disease progresses. And if you ask how many Citrobacter there are over time, when you give them to this mice, really you start to see about by day 3. And by day 7, the number of Citrobacter is maximal. And days 7 to 14 is when you see the most disease effects. You see a lot of inflammation, et cetera, and then the diarrheal type effects. And then the host acquired immune system, T and B cells kick in, and they basically get rid of the Citrobacter, they clear it out, and then it returns to normal, and basically Citrobacter is gone. Now that's what you normally see during an infection, where you basically you get the organism, the pathogen, you then get sick, your immune system comes in and fights it off, then you get better again. So here we have a natural infection cycle. And the real key question is, well, what happens to the microbiota as the mouse's intestine goes through this insult and the resolution of the disease? So this is what the microbiota of a C57 black 6 mouse looks like. So you can take the bacteria out, put it on a filter, and just stain the DNA, and what do you see? Well, you see many diverse species, and really they fall into 2 major shapes, these long rod-like structures through here, as well as these small sort of little round dot-like things here. So this is normal microbiota. You can see the diversity here that you've already heard about. And the question becomes, what happens when you infect them and give them diarrhea? So if you think about this, you think, well, what should happen? You can predict that, well, they're going to get diarrhea, so there should be a lot of things expelled, so the numbers should go down. That would make sense. And we'd always worked on this pathogen, and we always thought from a pathogen's point of view. So I assumed the pathogen would completely take over. That's pretty much all we'd see when you did this. Well, so here's before, and the next one is after. So there's after. Let's go back to before one more time. We then infect with Citrobacter 7 days into it, and this is what it looks like. Well, what do you see? First of all, you see less. Okay, that makes sense. But what you also see is you see a heck of a lot of these rods and very few of these little round things here. That tells us there's a change in the distribution of the species during this infection. And then the third thing you see is that this has also been labeled red for Citrobacter. And if you look really close, you'll see one here and one here. Yet this is at the height of the disease. So obviously the bacterium doesn't take the whole community over. It's present, obviously, but it causes some marked changes in that. So once we saw this, we were then able to start to quantitate these types of things. And when you compare the blue uninfected versus the red infected, if you just take a look at total numbers by 7 days, you can see that there's a massive drop, 80-90% decrease in the total number of bacteria. And this is the number of Citrobacter in the mouse at the time. And then as it wanes back to basically losing all Citrobacter, this then comes back up to the normal levels. So inflammation and infection is obviously having something to do on the total number of bacteria. Well, this is a few years ago, so we used fish to characterize this, and we used probes to the bacteroidetes, the proteobacteria, the firmicutes, and asked what actually is changing in here. And this is sort of what unaffected mice look like in the distribution of the bacteroidetes versus the firmicutes. By day 3, we're seeing a big change in here, and the proteobacteria includes Citrobacter, so red, basically. This represents the infectious agent coming up here, and by day 7, you can see there's much of it. But then the T and B cells come in to get rid of Citrobacter. You then start to change the distribution, and then it kind of gets back to quite similar to the way it does. So there's some kind of refractory pressure to shift the microbiota back to the way it was before the infection. So then we started to query, well, what's actually causing these types of changes? Is it the pathogen, or is it the host response to the pathogen? So these things cause massive intestinal inflammation. So we thought, okay, well, let's trigger intestinal inflammation by a non-pathogenic mechanism. Let's add this chemical, DSS, which basically causes openings in the gut and then causes intestinal inflammation by a chemical, and this is often used as a model of colitis, et cetera, et cetera. So when you trigger just inflammation with this chemical, again, you see a decrease in the total number of bacteria when you add the chemical. So intestinal inflammation itself seems to be enough to cause this decrease in this number. And then we had a look at what actually happens in terms of the shifts of the population. And remarkably, we see a profound increase in the proteobacteria. Now remember, there's no Citrobacter there, but the other proteobacteria, such as E. coli, they come roaring up when you trigger this intestinal inflammation, as opposed to control here. So that then started us on the concept that, well, maybe the microbe comes in, and this is a pathogen, but it actually triggers inflammation. So then the host then undergoes this gut inflammation, then shifts the microbiota and does something to that population, so it's a more favorable environment for the pathogen and its close relatives, such as E. coli, to then proliferate, and these things then proliferate and do well in here, and they grow until the host comes in and really attacks us with the acquired system and then kicks it out again. So that's where we started, and that's now led on. I'll tell you some more stuff where this is all going, because what we're starting to realize is microbiota do play a profound effect on this whole infectious process. So many years ago, when Bruce Valence was a postdoc in the lab, we took every mouse we could get a hold of, mouse type, and then infected it with Citrobacter. And like you would predict, you'd see some mice are resistant and some are more susceptible to infection. So if you take these kinds of mice here, and I change Swiss and these others here, these basically don't die due to Citrobacter. This is % survival. Yet if you take HEJ mice, these things, they will succumb to this. So these are susceptible mice, and these are resistant mice. And you see this again and again in infectious processes. And for years, people have been trying to map what mouse genes give you resistance to this disease. And there's some infectious agents like Salmonella. There's a great gene, the NRAM gene in mice, that really does determine, a mouse gene really does determine susceptibility. But nobody's been able to determine the host susceptibility to Citrobacter. And so then we decided to do something a bit wild and crazy. So maybe it's not the mouse gene that's doing it. Maybe it's the microbiota composition that may be dictating this specificity to this infection. So then Angeli Virchow and Ben Willing, when they were in the lab, they then basically started to do some, for lack of a better term, fecal transfer experiments. So what we're going to do is we're going to take the feces from a resistant animal, in this case NIH animal, and we're going to give it to a mouse that's susceptible. This mouse has been pre-treated with an antibiotic to get rid of its most microbiota. So the idea is put these, quote, resistant feces in here, and then see if we can then convert these to a more resistant phenotype. We'll also have a look at what happens. Can we actually fecally transfer these things and make these mice look more like the donor rather than recipient type microbiota? So that was the experiment, and we set about to do this. And what we found is that, yes, indeed, you can really drastically shift the feces, do these fecal transfer experiments. And this is what the NIH mouse distribution looks like. This is what the HEJ looks like. And then if you then reconstitute this HEJ mouse with the NIH feces, you can then make it look more like an NIH mouse than the HEJ itself. It's not perfect, obviously, but we've really drastically shifted it, making it much more like this. And that's what this dendrogram here is showing, basically that the HEJs that have been reconstituted with NIH feces, they cluster right nicely in between all the NIH mice and their relatedness, compared to the HEJs themselves or HEJs reconstituted with their HEJ feces. So that's great. That means we can make it look like more like an NIH mouse. Well, what happens when you infect? And this is where we're really surprised and really, I think, quite remarkably looking what happened. So if we just look at what mice survive or die, that's probably the most basic way of looking at this infectious process, that if you have HEJ mice reconstituted with their own feces, this is a susceptible mouse, it should die, and yes, indeed, it dies during infection. Yet, if we take these mice that always die and put in NIH feces, we now get huge survival rates. So we drastically altered the outcome of host susceptibility just by changing the feces. We didn't change the pathogen, we didn't change the mouse, just by the feces. And there's other ways of quantitating. You include weight loss, for example, and the HEJs with their own feces, they lose weight just before they die, whereas the HEJs with the NIH, they do just fine. And if you look at bacterial counts, you look at where Citrobacter is, basically the HEJ mice with the resistant feces in them, you just don't see the bacteria there at all compared to those that are getting sick. So this is, I think, a remarkable concept that we can actually dictate host susceptibility just by altering the microbiota. Well, if that's true, we should be able to do the reciprocal experiment and make resistant mice more susceptible if we put in the susceptible feces into these things. And that's what this is really just showing again here, is that you can see if you take NIH reconstituted with their own feces, they're resistant in terms of weight. If you put in the susceptible feces into these things, they now lose weight. They get sick, they have much more pathology, and they have much higher Citrobacter counts in there. So it works both ways. You can indicate both resistance and susceptibility just by shifting the microbiota of these animals. So what's going on? What's the mechanism? That's obviously what we're all quite interested in. And Ben has now gone on to probe what actually happens here. And one of the things, notice we're looking through various cytokines, et cetera. There's a cytokine called IL-22. And it was very high in NIH mice and very low in the susceptible mice here. And when you constituted the HEJ mice with the NIH feces, again, you got very high levels of IL-22. Now, IL-22 is a cytokine that regulates various antimicrobial peptides, including reg3 gamma and reg3 beta. And we measured those things. What we saw is we saw a really high level of this reg3 beta expression levels in both NIH and the HEJ reconstituted with NIH that was decreased here. Now, these reg3s are thought to be evolved, produced at the mucus layer, and they're thought to interact and basically kill off various microbes and alter the microbe distribution and evolve in host defense to actual disease. And so when you actually stain for this thing, are we seeing this stuff here? The answer is yes. You can see it staining quite nicely in these things here. So if that's true, then let's do the reciprocal experiment. And what happens when we put the susceptible feces into resistant mice? You predict that we should see less IL-22 because that was linked with the resistance, and that's exactly what we see. The IL-22 goes down when we put in this HEJ feces to that. And then to really nail it, what Ben and Angeli did was basically come in with blocking antibodies to IL-22, and they showed that if you injected anti-IL-22 antibodies into animals, you could decrease, compared to an isotype decrease the amount of the reg3 beta expression in this case. And you can also affect the susceptibility to animals, which is shown here. So basically, if you take the HEJ mice, susceptible mice, resistant feces, but mop out IL-22, they're now more susceptible than that with a control isotype. Now, I'm not saying this is the complete mechanism, but it certainly gives us clues as one of the ways the shift in the microbiota is affecting the whole outcome of the infectious process. I'm now going to talk a little bit about what happens when we shift the microbiota in terms of the using antibiotics and what actually that actually plays a big role in the susceptibility to infection. So the idea is we all know antibiotics kill microbes, and we've been blissfully using these things for years, but there's actually very few studies out there that have looked at the effects of antibiotics on microbiota distribution. And we reason that this would be a really good way of shifting the microbiota different ways, using different antibiotics to then actually see what happens to various things, including bacterial infections. So in this case, this is again the Citrobacter model. This is work done by Marta Werlotzka in the lab, a graduate student. And basically the idea was to give them just 2 different antibiotics at these doses for 4 days. Then we're going to affect with Citrobacter and just watch what happens. And we can study various parameters of this infectious process. Well, the first thing that Marta did was characterize what happens to the microbiota when you give the antibiotics. And the changes are profound in the distribution of the microbiota. You can see the various characterizations here. I'm not going to go into details. Obviously, they cluster together, depending on the metronidazole, the streptomycin, or the uninfected. And you can see just the drastic changes occurring in the microbiota when you give these things. Well, what happens when you affect with Citrobacter? Well, what we found is that when you treated with metronidazole, you had a much more severe infection with Citrobacter, but streptomycin had no effect on the Citrobacter infection. So let's say, look at this thing here. Here's uninfected, basically they're just fine, streptomycin, but you had metronidazole, you see a much more profound disease phenotype, a lot of infiltration, fluid, etc., bigger disease type, higher pathology score with metronidazole than if you didn't pre-treat when you gave Citrobacter. Well, then that begs the question, well, what's going on? Why can one antibiotic make these things more susceptible to infection, whereas another one can't? So one of the things we had to look at was the mucus thickness, as that we felt that might actually be playing a role in these whole things. So what Marta did was actually go and physically measure the thickness of the mucus in these different antibiotic treatments. And what she found is that when this is untreated, strep treated, or metronidazole treated, when you treat with metronidazole, the thickness of the mucus actually thins. And you can quantitate it here. And so for whatever reason, we're getting a thinning in the mucus when we treat with this metronidazole, which is somehow leading to an increased infection. Our first clue, our first reaction was, well, just that this is good. It's just due to the different bacterial burdens, and metronidazole is affecting the bacterial burdens and total number of bacteria that are present. But we found is that it's not a result of having more Citrobacter there. That doesn't seem to be the reason. The amount of Citrobacter seems to be very similar in the colon and the cecum or in the mesenteric lymph nodes. So it's not just due to the increased Citrobacter burden that's causing this decrease in mucus thickness. We also looked at several other things, and this is just to remind you that on the gut surface, goblet cells produce mucus. There's this tight inner mucus layer in which there are really no microbiota, and then the outer layer which has a lot of microbiota here. And we then started to measure things such as the mucus gene, mucin-2, to see what happens in terms of mucus production, and does that back up the idea of a thin mucus affecting it. And what we see is that, yes, indeed, that MUC2 expression goes down in the metronidazole, which has a thinner mucus layer, as does trefoil factor 3, which is related to this. It goes down, yet other controls didn't. So that's all pointing in the fact that somehow the microbiota is influencing the mucus thickness, which then translates into an increased susceptibility to disease. Sorry, you've already seen that one. So then we had to look at what's happening to the inflammatory cells during this infection, and does the mucus thickness actually affect or is it related to the inflammatory cells that are coming to the site of infection? And you can see these when you metronidazole treat, which is a thinner mucus layer, you have a large increase in macrophages, NK cells, etc., not T cells. And this was gut specific because you look, say, in the spleen, for example, there's no difference when you treat with these different antibiotics. So obviously there's a significant interplay there with the microbiota and the thickness of the mucus, which then translates to innate defenses that are impacting on this. This may be hard to see, but this is really just staining for the amount of Citrobacter present on these things, untreated with antibiotics versus metronidazole. And what I hope you can see is there's a whole bunch more yellow Citrobacter adhering here in days 4 and 6 than there are in the untreated control. Citrobacter is now able to get easier access to the intestinal barriers and or down to the intestinal surface, and seems to colonize better. And there's much more right down at the intestinal surface in these. I'm now going to tell you a related story in that it's a different bacterium that causes diarrheal type disease in mice, and that's Salmonella typhimurium. But really the concepts and the moral of the stories are very similar in terms of what I've just told you is Citrobacter, and this seems to be a theme that holds true for both Salmonella and the pathogenic E. coli related Citrobacter. So this story goes back a ways. Salmonella typhimurium was so named because it causes typhoid-like disease in mice. That's where it got its name. And we and all the other people in the world who have been studying Salmonella for many years used it as a model of typhoid fever. Now that's a very systemic disease, basically goes through the intestinal epithelium, doesn't really cause any diarrheal or enteric type infections, goes to the liver and spleen and lymph nodes, and it proliferates. It's a very systemic type disease. Well, a few years ago, Wolf Dietrich Hart and then our lab basically found that if you took a mouse and you gave them a high dose of streptomycin, an antibiotic, wipe out the microbiota, and then you gave them Salmonella, instead of this systemic disease, you now got a gastroenteritis. And that's the disease that humans get most of the time when you get infected with Salmonella typhimurium. So really, we are now accessing a mouse model of the more relevant human disease than we previously had, and as opposed to being a systemic typhoid type model disease. So that's neat too, because we really, we didn't change the Salmonella, we didn't change the mouse, we just gave them a dose of antibiotics, and boom, suddenly we've got a whole different disease. We no longer have typhoid fever, we have gastroenteritis, which is a very different disease involving intestinal inflammation, et cetera, et cetera. So knowing that, we decided to play around with antibiotics again, and we tried to use antibiotics instead of a high-dose strep, which knocks out about 90% of the total microbiota in the intestine. We tried to go with doses that physicians would prescribe, say, to patients, more relevant doses that are used all the time, that are thought not to affect the total number of bacteria too much. And this is going back to the similar shifts, what happens when you treat, in this case with streptomycin. And again, we're probing the bacteroidetes in blue, the yellow are the firmicutes, or the gamma proteobacteria in pink. And as you increasing doses of streptomycin, you can see drastic changes in the phyla distribution of the intestinal organisms. Now, this is streptomycin. I'm going to show you another one, tetracycline, which actually looks remarkably like streptomycin. So just keep in mind that strep and tet are similar. Again, very similar type distributions of these things. Then we also gave them vancomycin. Vancomycin really hits gram-positives, and it had a profound effect, a different way, in terms of the big shifts. And you can see you're now getting a lot of proteobacteria coming up with vancomycin. So strep and tet are similar, vancomycin is different. Let's infect them with Salmonella and see what happens. Well, what we found is that when we infect with Salmonella, so this is number of Salmonella in the organs, for example, if you infect with increasing doses of strep or vancomycin, we see much increased numbers of Salmonella and diseases I'll show you in a moment. Yet tetracycline, which was showing the same kind of phyla distributions as streptomycin, didn't get any more disease, perhaps even a little less bacteria here. And you see this both in the ceca and the colon. So what we think we're seeing is that for whatever reason, strep is perhaps targeting down at species level different organisms than tetracycline that then leads to this increased disease in the Salmonella counts. So, and I'm not going to show all the data, we've published all this now, but we now know that really it is increased disease. We've done the histopathology, et cetera, and there's real changes going on and real susceptibility to Salmonella during this infectious type process. So the big question is, okay, what bacteria are doing this? We give them different antibiotics, we get differences, and this is where the field is really heading and pushing. And of course, you can do the characterization, as Bill has nicely told you, of all these different microbes in there, and you can look for differences and similarities between the populations. And we're starting to, Rosanna Ferreira in the lab has been doing a lot of this, and we're starting to see organisms that are correlated. This correlates, for example, with increased protection to the disease, and these lactobacilli seem to be increased susceptibility to these disease. I don't want to go through this in details because really it's not all sorted out yet, but this is just kind of to indicate that we're now looking through the effects of all the different organs that are found in those that are infected versus those that are not, to see if we can't pin down species. Now, that's not to say I believe there's a single microbe that is going to make the difference between whether or not you get salmonella, enteric infections, or citrobacter. I think it's going to be a population, a community-based thing, and my guess is we'll probably generate a list of several species that seem to be coordinated with susceptibility to resistance to disease. And I think we're going to have to use a metagenomic type way of thinking about it. Okay, if this community has all these genes, what do they do that might actually be influencing the host intestinal inflammation and the outcome of the disease. So in the last few minutes, I want to tell you something we stumbled in fairly recently that I think has just profound effects on host physiology. And Kitano Antunes is a postdoc in the lab that was interested in what happens to the small molecules in the host when you infect with salmonella. So as you know, the power of mass spec these days is absolutely remarkable, and you can now take, say, organic extracts of tissues or material and then basically put it on a machine, and it will tell you the molecular mass down to many, many decimal points of everything that's there. And then you can then run that through a database search and then identify and pinpoint based on molecular weight analysis what those small molecules are. So the experiment in this case was we have our mouse, and we are either going to infect with salmonella or not. We're going to collect the feces in the liver, do an organic extract of that. We're going to then send it over to Victoria University close to us, and this lab here who has all the equipment to do basically metabolomics of this. And he then does an analysis of all the small molecules in there, and then they run through a database and they identify based on molecular weight what those molecules are. So this is absolutely remarkable, I found, because these are heat maps, for example, and here's several untreated versus infected mice of what happens, and each one of these represents a different metabolite in the feces and in the liver. And you can see that there's massive changes going on to all the small metabolites. And these are the molecules that basically run the body. These are all the hormones, the coursinoids, the arachidonic acids, cholesterol, et cetera, et cetera. And basically this is just the data going what we actually found. I think, well, let's just have a look at these numbers here. These are the percent of the total that are actually changed in the feces or the liver. So we're talking, 60% plus of all the small molecule host metabolites that we could identify are being affected during this infection. What are they? You're probably not going to be able to read this, but basically there are a whole bunch of different things. There's arachidonic acids, for example. There's sterile metabolism, all sorts of different metabolic pathways that are being affected. And so once we saw this, we said, okay, obviously it was pretty much overwhelming. Let's just pick a few of these and then just try and see, is it real, and are these changes we're seeing in decreasing after infection or increasing after infection real, and are they related to the infectious process? This is a nice blurry slide that's basically really cholesterol metabolism and biosynthesis. So cholesterol is up here, and I won't go through the pathway, but basically if it's red, it was changed in the feces. If it's green, it was changed in the liver. If it's blue, it was changed in both of them. The entire cholesterol pathway is being affected by this infection, and just drastic changes going on in the whole metabolic process. So obviously it's having profound effects on that area. Another one we dealt with are the coursinoids, and we found changes in those things. And the nice thing about this is that there are hints in the literature that these things are involved in salmonella disease, and you can see some of the papers over the years that indicate that these coursinoids and arachidonic acids, et cetera, are involved in the whole salmonella infection. So just to finish up now, I think, and I didn't tell you about the effect of antibiotics on metabolites, but that too has profound effects. About 80% of the host metabolites are affected by antibiotic treatment. So there's obviously major effects going on due to the gut microbe in the host metabolomics that are going on here. So just to sort of sum up the major highlights I wanted to tell you about is that we think Citrobacter comes in, we think it triggers inflammation, which is a bizarre concept from a pathogen, but that inflammation then changes the gut microbiota to a more favorable environment that Citrobacter can then proliferate in, and it basically makes it easier for it to then survive and work in there. I told you that in case at least of Citrobacter, my guess is this will hopefully come out in many different pathogens. The idea of host susceptibility, we've always thought that that's the host genes, not, you know, when 100 people go to a wedding and eat the potato salad and 20 get sick and 80 don't, everyone says, oh, they just have different genes. Well, I would argue that based on some of the stuff we're finding in the microbiota in many cases also plays a role in this host susceptibility. And when we talk about this, we have to include the microbiota in what is a host or a superorganism. I showed you the microbiota was heavily affected, or when you change microbiota with antibiotics, you affect mucus thickness, and mucus thickness, as you know, is a key defense against infections. And so just by shifting the microbiota, we affect mucus thickness, which then basically affects the Citrobacter infection capacity. I've showed you several times that different antibiotics affect microbiota composition very differently. And really, there's virtually nothing in the literature about the effects of antibiotics. And well, we saw some hints at asthma in one of the earlier talks this morning. But the idea of when people are prescribing antibiotics, there's never any thought to what does that do to the microbiota that might actually have long-term effects in some of these other things we're finding these microbiota are impacting on. And I showed you the concept with salmonella that both streptomycin and vancomycin, but not tetracycline, seem to make these mice much more susceptible to infection. Even when we don't change the total number of microbes in the gut, we just shift them. And so obviously there's some serious interactions going on there. And then I concluded with the effect of the infection on the host metabolome, and I think this is just a tip of the iceberg of what's going on physiologically, because these are all, as I said, the signaling molecules that basically make cells talk to each other, make organs work, etc. And I think this will be an area that will be really rich for discovery in the future. So with that, I say thank you. I want to thank the people that did this. Claudia Lupp got this whole thing started in our lab several years ago. Marta did the work on the Citrobacter mucus thickness. Ina did the early work on the salmonella antibiotic effects. Angeli and Ben were the ones that did the host susceptibility story I told you about. Catano did the metabolomic study, and Rosanna did the characterization of the different species associated with salmonella, and then our mass spec was done by Christoph Borch. So with that, I say thank you very much.